To help you smooth sail with the diagrams is Diagram Dazzles. Today we are going to study the NCERT series diagram on the female gametophyte development. This series of diagrams I am going to partition into three. The first section is of course the Megasporogenesis, that is A. The second section, which you can see below, is B, and that represents the female gametophyte development. The third section, which is C, it is actually the formation of a mature embryo sac. So let's see how the diagrams appear. The Megasporogenesis is here with three diagrams, which is series wise, as you can see. The second one is the development of the female gametophyte, which represents the nuclear division and ending with the cell wall formation. So here it involves four sets of figures, and the last one, which could be also asked separately for full 5 marks, is a mature embryo sac or a fully developed female gametophyte. And with all this, you will have to do the labeling and the detailing as well. So let's start with the first one, that is Megasporogenesis. It is the formation of the megaspore. Within the nucellus, the megaspore mother cell differentiates, which is larger, and it undergoes the meiosis 1 to form two daughter cells, which are collectively referred as megaspore dyad. Again, it undergoes meiosis 2 to form the megaspore tetrad, which is linearly arranged. So let's start drawing. The first one, you will have to draw an elongated spindle-shaped cell. This represents the megaspore mother cell. Darken the outline and then go ahead with also the drawing of the other cells of the nucellus, which remain thin and rectangular. So first demarcate their outline for the cell wall and then complete them, showing them as the rectangular cells. So once you are done with this, remember some of the cells which are basal at the base, you will have to draw it slightly larger and polygonal. So this is how you represent the cells of the nucellus. The centrally positioned one is a megaspore mother cell. After that, you will have to draw the nucleus with the nucleolus for the nucellar cells. So draw them as a circle with the centrally positioned dot. You can darken this nucleus. Yes, once you've completed with the other cells, now go ahead with the central cell, that is a megaspore mother cell. Draw a central large nucleus with a darkened center that represents the nucleolus. Also surrounding it, put finer dots that represents the cytoplasm. Now the second figure here, you should begin with the spindle shaped cell that represents the megaspore dyad, and hence you will have to divide this one into two daughter cells, and each one represent the nucleus and the nucleolus. Darken this cell and fill the cytoplasm. Also draw the surrounding cells of the nucellus, which has to be drawn rectangular, thin, lighter, and they have to be filled with the nucleus and the nucleolus. Now coming on ahead with the third stage. The third stage represents the vital one because it comprises of the megaspore tetrad. Here the megaspore has completed its meiosis 2, and hence you will have to show linearly arranged 4 cells. So draw a spindle and divide it into 4 cells like this. Preferably the lowermost one should be larger. Fill it with cytoplasm and the nucleus, nucleolus. Also show the surrounding cells of the nucellus. So with this, we complete the first set of diagram, that is Megasporogenesis. Now the labeling. Importantly, you will have to represent the micropylar end where these changes happen, and then label the cells of the nucellus, the corresponding stages, the megaspore mother cell, the second stage megaspore dyad. The third is important, showing the linearly arranged cells of the megaspore tetrad. With this, our first set of diagrams is completed. Next, let us go to the second stage, that is development of the female gametophyte. This is said to be monosporic. Why? Because out of the 4 megaspores, 3 degenerate and only 1 remains functional. That functional megaspore gives rise to the entire embryo sac. It undergoes 3 nuclear divisions, that is 2, 4 and 8 nucleated stage, and the 4th stage, as you can see here, involves the cell wall formation. So let's start. The first one, you will have to draw a spindle shaped cell with 2 nucleus as shown at the 2 ends, And the next one is a 4 nucleated stage. That is because from the 2 nucleated stage the cells undergo mitosis without the cell wall formation. And the third figure represents the 8 nucleated stage. So here there are 4 nucleus at the corresponding ends as it is shown. The fourth figure is very important because now you will have to slightly increase the size of the cell, and here you will have to reorient the nucleus. Here as it is represented, you can see that the nucleus start moving towards the center from each of the ends. So this forms the polar nuclei. For the rest of them, there are 3 at each pole. So 3 at the micropylar pole, 3 at the chalazal pole, and 2 at the center. Now the crucial stage appears, that is formation of the cell wall. The cell wall for the synergids, you will have to show it slightly in the dotted pattern, draw it very light as it is shown. So this is a kind of elongated cell which is formed, and then of course the central one, that is the egg, you will have to darken the cell wall. So this is the development of the female gametophyte. It involves a 2 nucleate stage, the 4 nucleate stage, and then the 8 nucleate stage with the corresponding last figure representing the cell wall formation. Before we go ahead, just remember this particular highlighted region represents the micropylar end, which is reverted in the final diagram, that is a mature embryo sac. So you will have to remember the inversion of the position. The micropylar end has to be drawn on the lower side and the chalazal end at the upper end. So to begin with, draw a oval shaped outline and demarcate the T cells, that is for the antipodals. So also represent the nucleus and the nucleolus. At the lower end, draw a heart-shaped diagram that would represent the egg apparatus, the synergids in specific. At the background, draw the egg cell. Now when you go ahead, remember certain associations. So draw 2 eye-like structures and then the ovals representing the vacuole. And then also for the egg cell, draw the nucleolus. There are small filiform apparatus which you can see, which is drawn at the base. So when you invert it, it would appear like the face of an animal. Yes, so remember this association. And finally, you will also have to represent the last and the important nuclei, that is the polar nuclei. Draw them slightly large, centrally placed, and also show the outline for the central cell. So this you will have to draw a parallel line running through the outline and dot it gently. So this represents the central cell. Now coming on to the labeling, the micropylar end and the chalazal end. With this, we'll start the labeling. Chalazal end has antipodals, 3 in number. The large space is to be represented as the central cell, the 2 polar nuclei with the center, and then the next one is the egg apparatus comprising of the egg synergid and the filiform apparatus. So this completes the mature embryo sac. So you learnt it. It's an elaborate diagram but an easy one. Yes, try it and then like and subscribe. Thank you for watching.